Yeah, it's really great to be here. I want to thank the BNI for hosting this. And I think I was just kind of reminiscing. I was here in Santiago. It was about, I think maybe, was it 5, 6, 7 years ago for another related, the Chilean-German consortium. But I had a great time. And I have to confess that a lot of the slides that I'm actually giving in the first part of this talk are similar because it's kind of not just a review, but it really is a story, kind of a trajectory of how the work started and where it's led to. In the end, I may actually include a slide I pulled off of a recent review that we put together, and it tries to bring together some of the neurophysiology and some of the sort of high-level thinking about brain function as an artificial network model of what we think goes on during these kind of conscious and unconscious states, role of sleep, the involvement of the specific kind of coding properties that I'm going to talk about, and the ultimate processing of memory, which I think contributes to our ability to understand the world, and that is sort of from sleep to general intelligence. That's sort of the trajectory. And so maybe by the end, after the second talk, we can talk about the sort of the big picture. But before we get to the big picture, we have to get to the small picture. And the small picture starts with this brain here, the rat brain, where you can see the overlying cortex removed to reveal the hippocampus. So what I'm going to be talking about is activity in the hippocampus during 3 primary states. One, active state of attentive awareness. This is during locomotion, and we've heard in some of the other talks, discussion of the sleep states, the resting state and engagement of the default mode network, and then the active attentive state. And so in the rodent, the active attentive state, it tends to be correlated with locomotion and the kinds of tasks that I'm going to talk about, that is in spatial navigation. One of the reasons for studying spatial navigation memory is that in rodents as well as in humans, the hippocampus has been implicated in navigational spatial memory, as well as episodic memory in humans. And so the question is, what's the connection between the critical role of the hippocampus and these 2 forms of memory, navigation in space, formation of episodic, sequential, autobiographically grounded events? And so a basic working hypothesis is that there's a critical function that the brain must perform in serving this sort of general need of general intelligence, and that is to build, understand, and execute models of the world. And that need is to understand, ultimately, structure of causality. Why do things happen? What are the causal agents that lead to those things happening? And how can we engage or direct those agents to make things happen that we want? And so forming a causal model first requires that we understand temporal structure, temporal ordering of events, capturing those temporally ordered events, evaluating them, and then turning them into a model that we can use some generative form to make predictions, to run simulations, to do all of the things that we believe underlie general intelligence. So the first step is, well, how do we capture time-sequenced events? And again, this is going to be largely just sort of a review of hippocampus, work that we've done trying to understand this process. And of course, the approach is multiple electrode extracellular electrophysiology. Drop electrodes into the space around neurons in the hippocampus, put them into a large array so we can implant a large number of these electrodes. Back in the day when Pedro was in Arizona, and we kind of applied this going from the rodent to cat primate. The idea was that this was an approach that would allow us to understand coding at the finest level of functional granularity, and that is spiking activity of populations of individual neurons, with the idea that information in the brain is going to be represented by patterns across neurons, and that by recording spiking activity from these extracellular electrodes, this is showing the raw data, we could construct profiles of spiking amplitudes across these 4 channels that would allow us to simultaneously record on the order of 100, maybe a few hundred neurons. And some of that early work looking at the coding of space using this approach in a little simple environment like this, a rat running around, gave us this picture of the hippocampal spatial code, which of course John O'Keefe, along with Edvard and Maybrit Moser, were given the Nobel Prize in 2014 for this discovery of spatial representation of the hippocampus. Now, I always like to point out that the tagline for the Nobel Prize has got to be dumbest tagline ever given for a Nobel Prize, and that is that the hippocampus has the brain's GPS. This has got nothing to do with localization in space. That was not really the compelling insight that O'Keefe and the Mosers actually made. What was compelling was that there was a representation of space that was somehow intimately linked to memory. And so understanding what is that connection between the properties that we see in cells in space and the role in memory. And just a general, just kind of a seed to plant in your mind is you can think about space as having 2 properties. One, the GPS property that we kind of, that the Nobel Prize Committee decided to promote, is this idea of a map. And that is that there's a layout of locations, and the relative relationships, the distances, the angles, can be calculated, and that's useful because if you have a space, a topographic space, you can do spatial inference. You can calculate shortcuts. You can actually compute on a map. But there's another representation of space, one that we're actually more familiar with and tend to use more frequently, and that's a topological representation. You can think of it as the representation of routes. That is the connection. When we're sort of thinking about how we get to some place, we generally don't bring up a topographic map, do calculations on that map, and then execute paths. What we really think about is we think about waypoints. We think about getting from one point to the next to the next. And so the idea of routes or topology, that is the connectedness of locations. And you can think of topology as being captured, in a sense, by the hippocampus, topography by the input in the entorhinal cortex. So you think about these 2 kind of spatial representations. And why topology is going to be important is that the topological representation is the way we experience space. We experience space as a series of connected locations. And this relates to this need of the hippocampus to capture temporally ordered events. So the topology of space is captured by temporally sequenced experience in space. And if you go from that, this is this sort of average map of firing rate activity of individual cells. This is 80 cells in which the color indicates the mean firing rate. And so what you see here, the representation of place cells, different cells, this cell, for instance, firing the center of the box, this one the upper corner, many cells silent, some of these cells seemingly active over the entire environment. These are inhibitory cells. These are the primary pyramidal cells of the hippocampus. So this just kind of captures the static picture of activity in the hippocampus during spatial navigation. And then this shows something that's a little bit closer to the raw data, and this is spiking activity at each point is an action potential during navigation of 2 linear tracks. The animal runs back and forth on these little tracks. And the cells are color-coded, yellow cell, red cell, green cell. And one thing that you note about this representation is you can see how the individual spikes are actually emitted. And I'll usually do my my rendition of brain activity as the animal runs through here. You hear this kind of popping, this kind of pop, 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 pop. Cells begin to fire. The firing is restricted to this region of space. But in an apparatus like this, these cells display another property, which is more related to the topological representation, and that is they don't just fire in this location, they fire in this location in a given direction. So the yellow cell will fire when the animal runs through in this direction, pop, 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 but not when it runs through in this direction. So the fact that they are directional means that there's going to be a different pattern when the animal runs along this path. It'll be yellow, red, green. When it turns around, it runs back, going through the same locations, but in a different order. there's a different sequence, a different set of cells. So the hippocampus already is demonstrating its capacity to capture different sequences in different patterns. But again, this is just the static picture. I have a movie which, I'll just. I'll fill in the audio. So when the animal's running, what you see here, so this is like the raw data, the blue cell. You hear this rhythmic modulation, the theta rhythm, which modulates the activity of these cells. And here you see the place cells, blue cell firing here, light blue cell firing here. And this is the data as it's coming out. Oh, there we go. There's some audio. Perfect. Thank you. If you listen to it, if you get the volume up a little bit, you may even be able to hear the. But this is just to illustrate that the property of spatial firing is incredibly robust. If you have a good ear, you can hear the rhythmic modulation as well. So here you're seeing raw data. This has not been processed. It's not been averaged. This is as it's coming out of the animal. You can already see the spatial specificity. But you notice there's some locations here. For instance, here, the spatial specificity seems to go away. Lots of cells firing, blue cell, red cell, yellow cell. I mean, there are locations, for instance, here and here, where that spatial specificity seems to go away. Those correspond to locations where behavioral state changes, and that is this rhythmic modulation associated with active attention. You could say encoding of information switches very rapidly into this inattentive mode, quiet wakeful mode, which is indicated by the presence of what are referred to as sharp wave ripple events. Transient bursts, about 100 milliseconds in duration, of elevated excitability, lots of cells firing. And it's during that state where you see the loss of the place code. But if we use another approach to illustrate this data, instead of just showing the raw data, what I'm going to do is I'm going to take the same data, but instead of showing the spiking, I'm going to show you the result of this Bayesian decoder. And that is, we're going to take the spiking activity and we're going to guess what's the most likely location the animal would have to be in order to produce that pattern of activity. So for instance, if the animal is here and if you hear the blue cell firing, you might guess, oh, the animal is probably here. And that is, you're computing the probability of being in a location given the current pattern of activity. And it's referred to as a Bayesian decoder because you can think what we're using the probability of spiking given the location. That's the classic, a receptive field is basically a plot of firing rate as a function of some kind of input stimulus dimension. Could be the angle of a bar, the size of a dot, the location of the animal. And so we compute firing as a function of input. We can invert that using Bayes' rule to compute the probability of an input given the activity of cells. So it's the same data. You can see the same data here, only now that probability is going to be reflected as a triangle, where the size of the triangle is the probability. Now remember, there's a probability estimate for every location on this track. It's just that the only place where the probability is high is basically where the animal is. That's the place code. So you notice when the animal's running, the place code tracks the animal's position. When the animal stops, and you get those bursts, you notice there's a lot of spiking, but there are also triangles on the track, and that is the code represents locations, just not the current location of the animal. So again, the two states, active locomotion, the code tracks the animal. When the animal stops, the code jumps around to remote locations on the track. So we can use this decoding approach, same thing, only now apply it during different states. So here we've taken the animal off, completely off the track. It's sitting in a little box. Here it is asleep. It's not actually on the track here. It's in a little separate space. But we can apply the same decoding analysis. And so you can see when we decode activity during sleep, it corresponds to, you notice that there's a sequence of triangles that run along the track here. And I'll go into a little bit more detail on this. During these inattentive states that would include sleep and quiet wakefulness, the hippocampus re-expresses spatial sequential code that maps onto trajectories on the track. And one thing, if you look at that particular example that I gave, you notice that the triangles seem to run backward in time. And that illustrates another property of this sequence memory reactivation. That is, it can run in either the time forward or time reversed fashion. So it's kind of like during wakefulness, during the theta rhythm, it's like you have, I use the VCR example. Nobody has a VCR anymore. But anyway, you record. I suppose, even DVDs, nobody has those anymore. But you're taking a record of events, a stream of inputs over time, and then once you have that record, you can go back and revisit it. You can rewind it, you can run it, in forward or backward at different speeds. And the other thing that you can do, which is going to be critical when we kind of think about the functions of these attentive, inattentive, sleep, conscious, unconscious states, is that you can edit it. And that's the critical thing, that you're not restricted to processing what actually came in, but you can take it and recompose, take out relevant elements, combine them with other elements in order to construct what we would think of as a more useful model of experience of events derived from the actual record of those events. So the objective is, you think of the brain as to build a model. You build a model by trying to incorporate experience. But ultimately, the model is a selective reflection of past experience. So there are going to be these sort of three elements that I'm going to talk about during these states. One is sequential acquisition, and that is capturing sequences of events in these neural codes that can be reactivated during these sleep states, sleep and quiet wakeful states. The compositionality, and that is the idea that these sequences are actually broken up into smaller chunks, where the chunks themselves can be reassembled in some way. And then the regulation of time, of speed and direction, and how those then contribute to memory and learning processes that we think go on during sleep and quiet wakefulness. And so this is just an illustration of the general states themselves as reflected in local field potential, or EEG recordings, as taken from Yuri Busaki, where you see the two primary modes of electrical activity expressed in the hippocampus and during active locomotion, or you could say encoding, or active intensive engagement in the world. This is the theta rhythmic mode. When an animal stops within about half a second, there's a transition into this sharp wave ripple mode. This was, I use this nomenclature. Back in the day, these events were actually referred to as large irregular activity. And so large irregular activity in the EEG were these spikes that had a large, reflected a large deflection in the extracellular voltage with an irregular or aperiodic structure. So it's just, it was like unstructured activity during quiet wakefulness and sleep. Upon more careful inspection, Yuri Busaki identified high-frequency oscillations, or what he referred to as ripples, that would ride on top of these large deflections. Hence, the term sharp wave is the sharp wave, and then ripples. And when we look at one of the properties, so this is during locomotion, active acquisition, quiet wakefulness, and sleep. When we look during the theta rhythm, we see that there's another important property that O'Keefe also discovered in the early 1990s, and that is that the spikes are phase locked to the theta oscillation. But they're not just phase locked to a particular phase. The phase actually changes as a function of time, and in this case, position as the animal moves, and as well as firing rate. And so this property, which O'Keefe termed phase precession, you can see here. you think of the animal as running along a track, it enters into a place field, and here you see the first spike emitted, low firing probability, and this spike occurs at the peak of the theta oscillation. As the firing rate increases and as the animal moves further into the field, the phase or the time of occurrence of the first spike relative to this peak moves earlier and earlier. And so this is the property of phase precession, which I'll show you later. But there's another thing that's illustrated here, and this is work by Thanos Siapas when he was in my lab. And if you look at spiking, and this is spiking in the hippocampus. This is spiking activity in medial prefrontal cortex, in infralimbic and prelimbic cortex that are connected to the hippocampus. And what you see here is that spiking activity in the prefrontal cortex is also locked to the hippocampal theta rhythm. So we'll see that this is actually relevant in trying to understand how theta-related activity gets communicated and used by other parts of the brain, and the idea that the theta rhythm serves as kind of a primary mechanism of communication through the process of selective coherence. Because essentially other brain regions tune into the theta oscillation when they want to listen to the hippocampus during both active encoding and active use of hippocampal memory. And this is the process of phase precession. And so this is a cartoon showing two properties of the hippocampus. One is the rhythmic modulation of excitability, as in this case represented by the red line, which I've termed inhibition. So you can think of inhibition being modulated by the theta rhythm in the hippocampus, and that excitation is going to come in this spatially modulated form. So I've kind of illustrated a ramp of excitation in blue on top of a rhythmic modulation of inhibition in red. And then I'm going to use a very simple biophysical rule that says the timing of a spike is going to be determined by the interaction between excitation and inhibition. So when excitation exceeds inhibition, that's when you get a spike. And the rule is the lower the excitation, the longer you have to wait till inhibition drops in order to produce a spike. So stronger excitation means earlier spiking. It's just a very simple idea. You could do this with a cell in a culture. You drive the cell harder, it spikes earlier. Weaker input, weaker excitation, longer latency to spike. And if you take that principle, magnitude to latency principle, and you apply it to inputs that look like this is what you would get. And that is that as the animal moves through the field, the phase or the timing of a spike gets earlier and earlier. But there's another property to this phase precession, which is interesting, and that is you notice that if I were to separate this into the late phase region, that's from 180 to 360 degrees, you notice there's a pretty good linear correlation here between position and phase. You can think of this as a temporal code that you can determine from the phase of these spikes the relative position of the animal. The early phase region here, if I didn't show you this up here and I just showed you this activity, you would say, oh, okay, there's just spiking activity. It's not really correlated with position. In other words, there's a weak linear correlation between phase and position in this region. This is the high firing rate region of the field. So you can think of this as reflecting a rate code, temporal code, rate code, separated by phase, early phase, late phase. And we're going to see, I won't talk about it in this lecture, but when I get to the next one, we're talking about conscious processing. We'll see that these two components of the hippocampal code can actually be dissociated. We can use optogenetic approaches to selectively manipulate activity in the early and late phases and determine that early phase activity is associated with the encoding of information. Late phase activity is associated with the retrieval of information. So you can think about their different codes in different phases. And that if we take this single cell model and we add in a second cell, this is where we get the most important property, in my mind, of the hippocampus. And that is the idea that using phase precession and then cells which are spatially organized, for instance, this place cell has a place field to the left of this place cell. So you think of spatially organized cells, cell 1, 2, and 3, using this principle of magnitude to latency. You notice place cell 1 is always going to fire before place cell 2 in each and every theta cycle. And that is phase precession in single cells translates into sequences in the theta oscillation, or what we refer to as theta sequences, in a population of cells. And that is that the hippocampus is actually creating a sequential code that's expressed every single theta cycle. And this is just an illustration of, again, raw data showing multiple cells, an animal walking along a track, and you see, as it walks, there are these sweeps where a series of cells will fire, suggesting a sequence. We can clearly demonstrate that by using the decoding methods that I showed you earlier, and that is if we decode the activity at a very fine time scale, in this case at a 20 millisecond time scale, and here the gray is actually used instead of those triangles and the size of the triangle to indicate the probability of being in a given location, given a particular pattern of activity. And so we can see that at a 20 millisecond resolution, the internal code actually sweeps from a position just behind the animal to just in front of the animal. And it does so 10 times a second with an effective velocity, which we'll see is going to be relevant, of about 5 to 10 meters per second. It's about 10 times the actual real-time speed of the animal. So it's brief, it's compressed, and it has this property of incorporating past, or linking past and future locations. So behind the animal, in front of the animal also corresponds to where did I come from and where am I going to? And if we just average all of these theta sequences, and that is that we sort of look at the average decoded position as a function of phase, this is the code. This is the hippocampal code for space. Again, it's not about location. This is the actual location of the animal. It's about relative location, the local topology, the sequence of locations from where I just came from to where I'm going to. So that's just the setup for the actual topic of the talk, and that's sleep and memory. But why this is important is you have to have this idea that it's really about time sequencing. Animals are encoding time sequenced information, and then during sleep, we're going to see how does that time sequenced information get re-expressed. So how are we doing in time in the sequence? Anybody keeping track? 15 minutes? We're okay? Good. So why look at sleep? We've had a lot of discussion of sleep, so I won't go into this too much. But one thing to point out is that in thinking about sleep and thinking about the different sleep stages, what I'll show you is that memory reactivation, which I'll illustrate, occurs during all of these stages of sleep. So if we want to think about memory reactivation, as I'll show it as an animal correlate of dreaming, we can think about dreaming actually occurring in all of these sleep states, but it has a very different form. And so we can kind of think about the different functions that might be served by REM and non-REM memory reactivation based on its structure. And so this is just an illustration of the experimental design that was run by a graduate student, Albert Lee, when he was in the lab many years ago. Very simple task. Animal sleeps, runs back and forth on a little maze, this little track, activating these place cells in a sequential fashion, then goes back to sleep. And this is actual sleep structure from one of these rats. Here we see in the period of sleep offset, where red indicates epochs of REM. REM epochs last on the order of 1 to 2 minutes. And you can see that as in humans, cats and other, basically all mammals that have a REM, non-REM structure, you see this loading of REM during periods of sleep offset. So there's more REM just before the animal wakes up than there is when the animal goes to sleep. So sleep onset tends to be more non-REM heavy. Sleep offset tends to be more REM heavy. And so the experiment involved looking primarily here at this period of non-REM sleep immediately following behavior. And what Albert found was, if you look at a series of place cells that are activated, in this case, in order, going from left to right, cells 1 to 8, what he found was when the animal would go to sleep, there were these periods where the same cells were activated in the same sequence, but that there was this temporal compression, that a sequence that took the animal 5 seconds to run through was re-expressed in about 150 milliseconds. And this occurred repeatedly. And when you looked at the occurrence of these sequential events with respect to the local field potential, what you found is that these sequential events occurred during the expression of these sharp wave ripple events. So sharp wave ripples were associated with sequence reactivation during non-REM sleep. And what's interesting is if you take that simple model that I described to you of encoding of sequential information in the theta oscillation, and that is this latency, magnitude to latency, stronger means earlier, and you apply it to the reactivation of memory, and that is I apply a state, and that is I give the hippocampus, I just give it an input, I clamp that input, and then I sweep inhibition, sweep excitability to unmask the magnitude of those inputs and translate them into latencies. The same mechanism that encodes sequences can be used to retrieve sequences. And, not that surprisingly, the time scale of sharp wave ripple events, a sharp wave ripple is about 80 to 100 milliseconds, and the mechanism of sharp wave ripple expression, that is disinhibition in the hippocampus, is basically the same as the theta rhythm. In fact, the medial septum, which regulates cholinergic and GABAergic input into the hippocampus, is responsible for regulating both the theta oscillation and sharp wave ripples. So this points to septal cholinergic systems as mediating the brain states and oscillations that are responsible for coding time sequence information, reactivating time sequence information in theta and non-theta states. Now, when we look at the other theta state that's associated with sleep, and that's REM sleep, again, you find very powerful cholinergic, septal cholinergic innervation of the hippocampus. So you can really think of the septum as sort of driving the whole show. It's driving the hippocampal states that are responsible for these different sorts of encoding and retrieval events. But how those events get used is going to be a function of the downstream recipients. So the hippocampus can basically just switch into these two modes: capture sequences, replay sequences. Their impact is going to be determined by what other brain areas are doing, what states those brain areas are in, which brings us back to the very interesting topics of these kind of altered states of consciousness, which you can think of all riding on top of these basic mechanisms. So you just get the hippocampus to do the same thing. It's just that you place these other cortical targets into different states to respond to that information differentially. So we're taking this, the idea that there's reactivation of sequential memory in the hippocampus during sharp wave ripple events. We can ask, what are the downstream recipients? And this was work by Dayun Ji when he was a postdoc in the lab looking in the visual cortex, running a simple task, which is the same kind of task that we've run, rat running in a maze, with one difference, and that is that there were prominent visual cues that were placed, proximal visual cues that were placed in the maze, that is textures on the floors and walls, and there were different inserts, different portions of the maze would have different textures. So this would have dots, maybe triangles, stars. So as the animal walked along the track, in addition to following a sequence of locations, it was also experience a sequence of visual cues. So you have sequential visual cortical activation, and this is illustrated here. These are two cells, one in the visual cortex, one in the hippocampus, showing that this visual cortical cell has an elevated firing rate in this portion of the maze. It looks kind of spatial. Here, the hippocampus is firing this part of the maze, but you would think of this as it's a visual spatial receptive field. It's firing here because the animal's in this location where there are the relevant visual cues. Now, one problem with this idea that the visual cortex is just responding to visual cues is that the cues are not unique to location. And as the same cue, the polka dot pattern, appears at multiple locations in the track, what does differentiate a pattern is, again, the sequence of visual cues that will be experienced. And so this visual cortical receptive field already reflects a property of visual cortical coding, which says it's not just the immediate cues that are relevant. It's something about either the sequence of cues or the sequence of locations that are associated with that cue that can determine the firing of these cells. And one way the idea that visual cortical cells are not just responding to the visual stimuli can be easily demonstrated by just doing one manipulation. That's just turn off the lights. You eliminate the visual cues, and what you find is that the firing of these cells is dramatically reduced by about tenfold, but the spatial specificity remains. So they fire in the same location, just with a lower rate. So it's this idea that visual cortical encoding is being laid on top of a foundation of spatial, perhaps hippocampally driven sequential information. And here you can see the sequential nature of visual cortical activity imposed by running on this track with visual cues in it, shown here. So this is activity of 8 or 9 cells as the animal runs one lap. And so you can see the visual cortical cells firing in order. Here, the hippocampal cells firing in order. And then animal goes to sleep. We look in non-REM sleep, and what you find is there are periods in which you find the same visual cortical cells firing with a structure that resembles the sequential structure during wakefulness. Same thing in the hippocampus. And if you do the analysis, you find, yes, indeed, there's visual cortical reactivation of the spatial sequences that are reflected in the hippocampus. So hippocampus replays a sequence of spatial locations. The visual cortex will replay the visual cortical sequence associated with that. But here, if we step back and look at the macroscopic structure of activity during non-REM sleep, here in the visual cortex and here in the hippocampus, we see something interesting. So this is just looking at the local field potential in the visual cortex, and then here in the hippocampus. In the hippocampus, you see activity dominated by these sharp wave ripple events. The sharp waves have been filtered out so you can see the ripples. The cortex, what you see is the kind of classic K-complex slow oscillation-like activity. So here you see the K-complexes associated with periods of sort of general quiescence. There's silence in the neocortex, which we would think of as being associated with down states in cortical pyramidal cells. Interleave with periods of elevated activity across populations of cells, which again we think of as being associated with upstates. So this is the slow oscillation in the cortex associated with corresponding activity in the hippocampus. What's interesting about the hippocampal activity is that hippocampal pyramidal cells do not possess intrinsic up-down state transition properties. They just don't. They don't have the kind of up-down state transition properties that are reflected in neocortical neurons because they don't have the right channels. So any kind of up-down state activity you see in the hippocampus must be driven by inputs. It's not being expressed intrinsically. You take these visual cortical neurons, you put them into a dish, and they'll spontaneously produce slow oscillations. You do the same thing with hippocampus, and you don't get any. So the fact that you see slow oscillation-like activity, and if you look at the latency structure here of the transitions, the upstate transitions, upstates in the visual cortex lead upstates in the hippocampus on average by about 80 milliseconds. So the idea is that visual cortex or neocortex turns on, the hippocampus responds by increasing activity, and then during one of these upstate-like periods, which we term frames, that's when you get sharp wave ripple events in the hippocampus. During sharp wave ripples is when you get sequence reactivation. And when you look at the relationship between sequence reactivation in the hippocampus and sequence reactivation in the visual cortex, what you find there is that the latency relationship is flipped around, and that is the hippocampus leads the visual cortex. So the visual cortex leads the hippocampus in initiating an upstate, but the hippocampus leads the visual cortex in driving the sequence. So visual cortex requests a sequence, hippocampus replays a sequence, visual cortex responds to that sequence. And that this process is then repeated. Now maybe when I get to the end, I'll put up this sort of a network model that captures that kind of process. What's interesting about a network model that involves this kind of bidirectional dialogue is that it corresponds to a class of network models, these so-called temporally recurrent Boltzmann machine-like models, which are capable of doing broad temporal statistical inference. As they kind of capture structure, that information is spread out over time. A good example of that is like just text or speech. In order to understand a word now, you kind of have to connect it up to words that were actually uttered in the past. That is, you have to be able to kind of connect things over time. And this back and forth like structure embedded in a network is able to capture that. It's able to find connections between things that were separated in time and then understand the overall meaning of that. Now you think about that in a topological model. Well, that's what navigation really is. It's like connecting up distant locations. I'm sitting here. How do I find my way to the bathroom? I have to connect up. I have to find the connections across temporally, just finding my way to connect, a word here in a sentence to another word in order to understand the meaning. You understand this can be a very broad computational principle. The use of this hippocampal neocortical dialogue to capture distributed temporal statistics in order to understand the meaning of a series of events, what we might think of as the context dependence of temporal experience. And so that's kind of captured here in the visual cortex. Now, the other thing that's interesting, and there have been a number of talks about this, and that is the idea that what this actually reflects is that during these non-REM reactivation states, the visual cortex is actually imagery. Animals are dreaming in this state. The visual cortex is it's not just active, it's active in a way that corresponds to the visual experience the animal actually had. We decode activity in the visual cortex. What we will decode is a sequence of visual stimuli along a path. Now, this idea that there's a relationship between the neocortex and the hippocampus, in which neocortex sensory areas are able to drive or initiate memory reactivation in the hippocampus, prompted Dan Bender, when he was in my lab, to run this experiment, and that is train an animal on a little task where it just runs back and forth along this track, with the additional contingency that the direction that the animal goes, starting from the center, is indicated by an auditory cue. The animal sits here, pokes his nose, and it gets one of these two auditory cues. There's frequency sweeps. This one, phew, wheep. Same frequencies, it's just the ordering of them. So there's the upward sweep, wheep, means go to the right. Phew, means go to the left. So the animal's just sitting here, pokes his nose. Here's one of those, and then goes to the corresponding side. And then the animal goes to sleep, take an animal off, put it in here, and then just randomly play these sounds. Wheep, phew, at a very low rate, about one every 5 or 10 seconds. So just very occasionally, and the stimuli themselves are only about 750 milliseconds in duration. And this just shows the activity, decoded activity. So we have hippocampal place cells. We can decode the position as the animal runs along the track. You have different cells, some place cells on the right, some place cells on the left. But the real question is, okay, the animal goes to sleep. So the animal's awake here, animal goes to sleep. If you kind of look in here, you can kind of see this frame-like structure being expressed here. And if we zoom in, we're going to ask, when we play one of these sounds, in this case, we play the left-hand sound, which is phew, phew, phew. And then we're just going to look at decode activity in the seconds that follow that. And what Dan found was when you play the left-hand sound, what happens is when you zoom in, for instance, in this little period here, which is like maybe 5 or 6 seconds after the cue, you find that the memory reactivation is actually biased toward the side that was associated with the sound. Play the left sound, it replays the left side of the maze. Play the right sound, you get the right side of the maze. That's just this. Yeah, play the left sound, activate the left side. Play the right sound, activate the right sound. But what's also interesting about this is that if you look at this bias, the ability to influence content, it persists. The cue is delivered here, 750 milliseconds. 5 to 10 seconds later, you're still seeing hippocampal memory being biased toward the side associated with the cue. how is this actually being maintained? Well, again, if we take this model that what's going on is cortex initiates, hippocampus replays, and then this stuff just kind of bounces around, it bounces back and forth. And in the process of going back and forth, the hippocampus and the neocortical dialogue are going through a process of refinement, trying to figure out what did that sound actually mean. You can think of this as a process of learning. So you can think of during sleep, there's a process of learning that goes on that involves the repeated reevaluation of previous behavioral contingencies. And this demonstration that there can be this kind of sleep bias and influence on learning was demonstrated in humans with a series of experiments, some early experiments by Jan Born using olfactory stimuli, and then more recent experiments done by Ken Paler using the same kind of auditory cueing protocol, which is quite striking. A simple task, card matching game, visual cues and locations. You have to remember where's the cat, where's the teapot. But in this task, Paler also played sounds. When you expose the teapot, you would hear a little whistle. You expose the cat, you hear a meow. And then during sleep, you would selectively play sounds associated with the cat or the teapot. And then when people would wake up, they would perform on this task. They would do better finding the cues that had, for instance, the cat, if the cat sound had been played during non-REM sleep. And so it's this idea that you have the cortex, you stimulate the cortex, this biases the hippocampus then replays, biases which memories get transferred to the cortex. And so there's learning, there's a sleep-dependent learning that goes on. And so that's in non-REM sleep. I'm just going to sort of briefly visit this question of what goes on during REM sleep, an experiment that's done by Kenway Louie when he was a graduate student in the lab. It's the same experiment. The difference here is the animal's running a little circular track, which you see is relevant because the nature of this task was when animals run on those tracks, each trial, it takes about an animal about 5 seconds to run from one side to the other. And it's just repeated back and forth. So the cycle time of the task is about 5 seconds. In this task, the task was to run from one location to another location 3/4 of the way around. Now, what's interesting about this is if the animal so the animal will go from here to here, the next 3/4 goes from here to here, the next 3/4 from here to here. So a full task cycle actually takes 4 of these trials. This task takes on the order of about 45 seconds to run. So if you look at the sequential structure of the task, and that is the activity of cells over time, you can see that the intrinsic period, the sequence that's expressed, is actually a sequence over, again, about 45 seconds to a minute. So we're looking at long sequences. And when we look during REM, the period is lasting about 1 to 2 minutes. What Kenway found is that if you look at the long time scale structure during REM, the long time scale structure seemed to be recapitulated during REM. And not only was the long time scale structure of play cell activity recapitulated during REM, I'll just skip this, this is just showing that it's actually reactivating what the animal experienced. Not only was the detailed structure of spatial locations recapitulated, but when the animal would run and stop during awake behavior, as I pointed out, there's this theta rhythm correlate. So the theta rhythm is associated with real locomotion. So here, during actual behavior, this is a spatial sequence associated with running, and then stopping, and then running again. So you can see if you look at the theta modulation of activity here, during running, lots of theta oscillation. Stopping, theta goes away, moving, theta's present again. When you decode the same sequence, the corresponding sequence during REM, what you see is the theta state is recapitulated. During REM, the animal is literally running. It is recapitulating, not just replaying the memories, it's actually recapitulating the behavioral states associated with it. It really is kind of a virtual simulation or recapitulation of experience. And this just illustrates. So when you look at REM, at run, you see this modulation of the theta oscillation. This is sort of the period of the task. Again, it's about 15 seconds for each trial. So you can see this kind of rhythmicity. And then you can also see, if you look in REM, there's also some modulation of the theta rhythm. So you think of REM, and often REM is characterized by elevated theta rhythm, elevated theta power. But what this illustrates is the elevated theta power, it's not just an increase in theta power. The theta power is the result of recapitulating behaviors or replaying behaviors that have theta power associated with them. So there's a structure to theta that you can decode, in a sense, to reveal the underlying content. So again, this is just REM. Basically, I think I'm maybe near the end. REM. So you have these long sequences in REM. It's approximately in real time. There was another interesting property that if you looked at the scaling, how long did a REM sequence last? And so it took, in this case, about 2 minutes of REM to replay about 1.5 minutes of run. And that is that there was a slight expansion. Things ran a little bit slower. What was interesting about that is the expansion factor was corresponded to the scaling in theta frequency. So theta ran a little bit slower during REM than it in run. And so the idea that the theta rhythm is actually the temporal metric. Theta runs faster, time runs faster. Because, what is time anyway? I mean, time, it's an internal construction. The argument here is that temporal metric is basically carried in the theta oscillation. So you want time to run faster, just speed up your theta rhythm. And so I think for this second, this is it. So sequence memory C encoded during active behavior, replayed in both the hippocampus and neocortex in non-REM sleep, and that we can bias that, and it also occurs in REM and non-REM, but with a very different structure. And that then will lead to the next talk, which I won't give right now, where I'll talk about some more recent stuff based on this, thinking about, again, how all these processes contribute to kind of extended learning and memory across these different wake and sleep states, awake and non-active wake states. All right.
Okay, so we're running a little bit late, so. So we have time for like 3 questions. It's okay? Yeah. 4 questions. Okay. Rodrigo?
Thank you very much for your talk. You just replayed all the stuff that I love from Hippocampus. I have a question though. How does the model of theta oscillation fits on humans or non-human primates, even bats?
Yeah, the bad stuff. So this is
because they don't have theta rhythm. They have like bouts of theta. So
yeah. So this brings up there's this sort of controversy: is the theta rhythm necessary for the kind of properties we see in the hippocampus? So I made an argument that theta rhythm contributes to an essential property of the hippocampus. This is sequential encoding. There were other models that posited that theta rhythm was actually responsible for the expression of place cells, grid cells, the kind of the fundamental behavioral correlates. And then Nakamura and the bat found that when you look at bats flying around or even kind of walking around, that bats had place cells, they had grid cells, but they didn't seem to have this persistent theta oscillation. And so this kind of called into question this whole class of models. Is theta really necessary? And then, of course, before this, there was the contention that theta oscillations were something really specific to the rodents, that you didn't see it in non-human primates. And humans, theta was weak. So is theta really that important? Maybe it's just, maybe it's just sort of an epiphenomenon. Maybe it's something that's, specific to rodents. I think that over the years, theta has emerged as something which is not rodent-specific. But what it does point out is it is very behaviorally specific, sort of behavioral, task specific. So in the rodent, the reason you see theta all the time is because animals are running and there's a locomotive correlate. In non-human primates, you can see it under certain conditions. So for instance, saccadic eye movement will be associated with the theta rhythm. So the way in which humans explore the world is different and more varied than in the rodent. And as I tried to argue, it's not just a kind of active engagement. It may be additionally that there's a demand on attentive encoding. So it's a mode of both attending, of both having a state in which inputs are changing and then being attentive to those changes. that's where you would see the theta oscillation. So it's kind of an active process. In the bat, the idea that you see theta, but it's in these short bursts, it wasn't necessarily associated with the kinds of things you might expect, and that is kind of active exploration of the sort of the auditory space, but it was present. And one thing about the model that I described is, even though it's expressed during the theta rhythm, it's not unique to theta, and that is any kind of oscillatory, variation in excitability will translate into exactly the same sort of properties. So you can have sequence encoding over delta, over gamma, and these things have been shown. Earl Miller, for instance, found this kind of sequential dependence in encoding in the prefrontal cortex associated with gamma in a kind of this was a Q sequencing task. And interestingly, he found that you could actually predict, it was like there was sort of an internal search, rapid search in this this kind of Q sequence space, which mapped on to phase. So it's the idea that if you have an input dimension where the value of that input is reflected in some kind of magnitude measure, then any time you start sweeping in it, you have some time-varying excitability, like time-varying inhibition, it's automatically going to give you sequences. And the only question is the magnitude actually related to some useful encoding dimension like time? And so my general contention is that it will be related if the magnitude is associated with like an integrator. So anytime you see ramps in activity in the brain, my argument is it is going to necessarily turn into sequences. So ramp encoding and firing rate is going to translate into sequential encoding in underlying sort of oscillatory dynamics. So not specific to theta, and theta is not specific to rodents. That would be my argument.
Here. Thank you, Matt, for a great talk. I have two questions. If I understood correctly, your model, this temporal sequence it's a sort of a temporal sequence,
right.
Which means that it may also depend on the speed. So would the rat will do this at an optimal speed, and if they change the speed, what would happen?
So that's the beauty of the simple model, this magnitude of latency model, the theta oscillation model, is it eliminates absolute time. It makes no difference how fast the animal goes through it. It just captures time order, because it's mapping space onto time order. And how fast the animal actually samples that those spatially ordered fields doesn't make any difference. That will just change the number of theta cycles over which you see the sequence. So what it's doing is it's transforming absolute time into relative time. It's creating time-ordered sequences out of temporal sequences.
But you need to match the visual spikes into the theta rhythm and the proper phase.
Now, mapping back into REM, now that's an interesting question. How do you get back real time? And so REM illustrates that the brain knows how to go back into behavioral time. So how does it know, what the time spent stopping, what the time spent running? How does it actually capture information about velocity? And so there's some interesting properties to these cells, which John O'Keefe, when he discovered phase precession, and that is that phase was correlated with position, his take on that was actually the hippocampal code is split into two pieces. One, a phase code for position and a rate code for velocity. This is what his original paper on phase precession with Michael Recchi in 91. That's what he argued, that the rate was actually was correlated with velocity. Now what's interesting is if you actually look at kind of rate and what that reflects in sort of excitability drive, stronger drive will also translate into slightly higher frequency. And we actually found this even like these sharp wave ripple, the replay events, that there's a correlate of instantaneous velocity, like gamma velocity, gamma frequency, and velocity. And so that you could extract, it's like, if I want to know how hard were these cells being driven, what was the magnitude of drive? And I think about the kind of, the local EI loop, pyramidal cell, local, sort of perisomatic, like, fast GABA A mediated inhibition and that model of gamma oscillations. If you drive the excitatory side of it harder, what you get is you'll get slight variation. It's an increase in gamma frequency. So if you could decode that, then you would be able to use that. You could say, oh, okay, stronger drive, higher higher velocity, less time. So you could use frequency as a metric for time, which I kind of also suggested in like the theta domain, where you see that scaling in sort of overall REM time. So that would be maybe it's not answering your question, but.
Hey, Mac, as usual, brilliant. So I was very much struck by the similarity with language, right? So we think that language is also a sequence of events, right? What is also interesting about language is that even though we speak in a linear fashion, we have syntax, which is a different form of sequencing, right? There is embedding. So do you have a sense of whether the hippocampus may play a role in those kind of structures as well, and not just the linear structures as you showed today, for instance, for spatial coding?
Yeah, so the idea, which I'll, again, the next part where I actually look at this sort of compositional structure, where there's this linear structure, but it's in the form of these short sequences. And so the idea is that the hippocampus captures linear temporal correlations, but only over a short time scale. And the reason for that is that's the time scale of sort of like high probability transition. When you really know, if I'm here, I am extremely confident where I was and where I will be about 3 seconds from now. And that's about the time window over which you see these sharp wave ripple theta sequences, sharp wave ripple sequences. Now, to actually go beyond that and try to make predictions or estimations that go beyond about 3 seconds, now what you have to have another mechanism for linking together sharp wave ripple events. It's the mechanism of sharp wave ripple bursts. And so this is where the idea is hippocampus can't do that alone. So now you have to go out and there's going to be this nested. So you have a structure in which you have nested oscillations with the fundamental unit being this kind of 3 second, sort of predictive, sequence encoding. So you can kind of, and then how you then arrange those shorter sequences into slightly longer sequences, which would be on the time scale. I think the next nesting is probably on the time scale, about the slow oscillation, about half a second to a second. And then there's going to be a scale beyond that in which you link across these frames of slow oscillations. And so this is, if you can kind of think about it, it's like the kind of letter, the word, the sentence. And so you're going to have, there's going to be an intrinsic nesting of time sequence structure reflected in the nesting, which interestingly, like Busaki, I mean, he had this, he had a paper, noting the kind of this sort of logarithmic structure to frequencies. And so you could think that's exactly the kind of structure that you would need if you were trying to do this kind of hierarchical embedding. Like, how can I even think about, temporal structure that spans an hour, a week, a year? Can't be linear. So, but if it's logarithmic, right, I just have this, it's very easy to span those sorts of time scales.
But are you thinking of the hippocampus as having all those windows of integration, or of different areas having different windows of integration?
Exactly. So it's the interaction between those areas that allows the hippocampal representation, which really is, it's just this little, for me, it's the theta sequence, the sharp wave ripple sequence. That's what the hippocampus knows about. That's all the hippocampus knows about. And it can be linked together through interactions with other structures. For instance, like the prefrontal cortex, which could have this much longer time scale through what we think of as like working memory delay activity. Just hold on to activity for, as long as it, the sort of a task period takes. That's kind of a task phase dependent embedding, which you can think of hippocampus, linking hippocampal sequences by prefrontal, delay activity. Hippocampus doesn't know anything about the temporal structure of the task. The prefrontal cortex knows, but the hippocampus has the detailed sequence encoding information that's required in order to figure out what actually went on in that defined task structure. Prefrontal cortex knows the structure. Hippocampus has all the details. By connecting the two, that's how you reconstitute sort of both task and content.
Last question.
I'm wondering, about where is the information coming from during the reactivation. I mean, for example, during the acquisition, we know that the entorhinal cortex is giving the information to the hippocampus, but what happens during sleep? And I'm wondering whether this is the same mechanism during awake sharp wave ripples, for example.
Yeah, I think that's it. As Thomas was pointing out, this kind of the multimodal, transmodal areas that provide the kind of convergent input into the hippocampus. That's likely what's driving activity during both states. But in the next talk, I'll talk about this kind of phase, this kind of phase segregation. And what's interesting about this phase segregation, where this phase procession is like a temporal code, late phase temporal code region, early phase rate coding. Those two phases actually correspond to different inputs. So there's a phase during which direct entorhinal input tends to hit CA1 in the hippocampus, and that's where you see most of the rate information. In the late phase is where you see CA3 information coming in. And CA3 is also getting it from the entorhinal cortex, but is running through some additional processing. So it's not just that you have like all this kind of high-level multimodal information converging on the hippocampus, and then the hippocampus is taking that and turning it into a sequence. There's a little bit more than that, and the little bit more is, if you kind of think about networks, there's sort of this basic function that artificial network can perform, and it's what's referred as autoencoding. Autoencoding is, how do I actually create models of the input world, right? You get a few samples from the world, and what you need to do is you need to build a model. You need to create what you really want are distributions. What are the distributions over inputs on that input space? And so an autoencoder will take a few samples and give you a distribution. Autoencoder means I build a model of the inputs. So I give it inputs, the input is the input, and the output is the input. It creates, but on the other end, it's a distribution. It's a probabilistic model. It doesn't just map the inputs back onto themselves. It maps the inputs back onto a set of distributions that might reflect that. And so you can think what you actually have, the hippocampus is very good at rapid learning, one-shot learning. What does that mean? One-shot learning doesn't mean I see this thing and I just capture this one thing. What I'm trying to do is I see one thing and I'm trying to extrapolate. I'm trying to turn one thing into a distribution of things. And so an idea is that what you have is the hippocampal, part of the hippocampal circuit is actually doing this sequential autoencoding. It's trying to take a sequence and then create a distribution of sequences. What are the sequences that, what does the sequence actually correspond to in the space of all the sequences? And I think that might be going on, for instance, in CA3. So you would have a CA3 autoencoder, which is essentially predicting. It's a prediction. Autoencoder is taking input and it's just making a prediction. What kind of things could I get? Another useful property of autoencoders is once you have this model of distribution, I can generate new things. So I give you an input, I build a model that's, a network that has that generates a distribution. I can generate other things like this to simulate things. And so the idea of simulation as creating instances from a single sample, which you then use to drive learning. I like to point out this is the basis of like the kind of contemporary artificial intelligence algorithms like AlphaGo, which beat the Korean Go champion. The method that AlphaGo employed was to essentially play itself, run simulated games. It did exactly this. It had this kind of autoencoding-like structure. You see a couple of games, you build models, and then you run through these simulations to actually learn the game. So this is the kind of thing that the hippocampus is probably doing, same process, build models, generate examples, and learn from them. And that's the phase dependence. So the circuitry in the hippocampus is doing more than just mapping onto sequences. The sequences are a reflection of some kind of sophisticated autoencoding in the service of building models that it can use to drive simulations, which it can then use to learn about, and solve problems in the world.
Thank you so much, Professor.
All right.